Hello, my name is Professor Hudson, and let's continue talking about DNA replication and how it ensures genetic constancy during cell division. DNA replication produces two DNA double helices, each with one original strand and one new strand. DNA replication begins with enzymes called DNA helicases that pull apart the parental DNA double helix at the hydrogen bonds between the complementary base pairs. A second strand of new DNA is synthesized along each separate strand by DNA polymerases, which pair free nucleotides with their complementary nucleotides on each separated strand. The two resulting DNA molecules have one old parental strand and one new strand. This is called semi-conservative replication. If no mistakes have been made, the bases of the new DNA double helices are identical to the base sequence of the parental DNA double helix. When DNA is replicating, as you've seen in some of the previous videos, new nucleotides are added to a growing chain. However, the enzymes involved in this must proofread the DNA as it's being made to ensure the number of errors is as low as possible. Remember, errors in the DNA are going to lead to mutations, which can be good or bad depending on the gene that they're in. Let's watch a video that illustrates how the DNA polymerase enzyme proofreads the DNA during DNA replication.
Although mistakes can happen during DNA replication, they are extraordinarily rare. A key reason for this is the proofreading function of DNA polymerase. Roughly 1 out of every 100,000 to 1 million bases attached to the 3' end of a growing DNA chain will be a mismatch, improperly base pairing with the complementary strand. When such a mismatch occurs, DNA chain elongation generally pauses. This pause allows spontaneous melting of the end of the DNA strand being synthesized, freeing the 3' end with the mismatched base to enter a 3' exonuclease site on the DNA polymerase. The exonuclease site catalyzes the removal of several nucleotides from the 3' end of the growing strand. Only the removal of the nucleotide carrying the mismatched base is shown here, though additional nucleotides would actually be removed. Following removal in the 3' to 5' direction, DNA polymerase can then resume its synthesis activity in the 5' to 3' direction. The proofreading function reduces the overall error rate to as low as about 1 in 100 million for DNA polymerase 3.
Let us review. Question 6. DNA replication takes place. Choose the correct answer. Question 7. If the left parental strand of the DNA molecule in the figure had a nucleotide sequence of T, A, T, G, G, C, A, what would be the resulting daughter sequence of the right parental strand? Choose the correct answer. We have now finished with the narrated PowerPoint lectures for Chapter 12, and now we will begin the narrated PowerPoint lectures for Chapter 13. And for Chapter 13, we will cover 13.1, how is the information in DNA used in a cell? 13.2, how is the information in a gene transcribed into RNA? And 13.3, how is the base sequence of messenger RNA translated into protein? Let us begin. The central dogma. In genetics, it is accepted that DNA can replicate to make copies of itself, or DNA can be transcribed to produce messenger RNA for making protein, and that messenger RNA can be translated into the protein necessary for building the organism. This is the flow of genetic information in all living organisms. This slide gives an overview of the central dogma. Information must be translated into action in order for a particular process to work. DNA contains the molecular blueprint of every cell. Proteins are building blocks and the foundation of the cell. Proteins also control cell shape, function, reproduction, and synthesis of biomolecules. Therefore, there must be a flow of information from DNA to protein. DNA provides instructions for protein synthesis via RNA intermediates. DNA information must be carried by an intermediate such as ribonucleic acid, aka RNA, from the nucleus to the cytoplasm. The DNA is in the nucleus, but it is too big to get out of the nucleus other than if the nucleus completely broke down, like it does during replication. So, cells will make a snapshot of the genes of interest in the form of messenger RNA. DNA in eukaryotes is kept in the nucleus. Therefore, this RNA must leave the nucleus, and it's small enough to do so, in order to go to the cytoplasm to make a protein. In prokaryotic cells, where there isn't a nucleus, protein can be made immediately as RNA is being made. Now let's talk about transcription, and we'll also talk about a little bit of background information on RNA. RNA differs structurally from DNA in three ways. RNA is usually single-stranded. RNA has the sugar ribose rather than deoxyribose. And RNA contains the nitrogenous base uracil instead of thymine. This figure lists some differences between DNA and RNA. Remember, DNA makes up the blueprint for the organism and is kept in the nucleus. Within the DNA is genes, and RNA is an intermediate, which is like a snapshot of genes that can be used for building a protein. Question 8. RNA is different from DNA because choose the correct answer. There are three types of RNA involved in the expression of genes. Messenger RNA is the primary one you hear discussed. This is the one that is the snapshot of the genes in the DNA. There are also some additional RNAs that play a role in making the protein. There's ribosomal RNA and transfer RNA. As mentioned, messenger RNA, as seen in figure here, part A, messenger RNA carries the DNA gene information to the ribosome. Codons, groups of 3 bases in messenger RNA, specify which amino acids will be incorporated into a protein. Transfer RNA brings amino acids to the ribosome, and the tRNA delivers amino acids to a ribosome where they are ultimately incorporated into a protein. Every cell synthesizes at least one unique type of RNA, and I should say tRNA. A group of 3 bases called an anticodon protrudes from each tRNA. So the tRNA, which stands for transfer RNA, will bring in the amino acid for making the protein. And it does this by matching its anticodon, which is a sequence of 3 nucleotides, with the codon, another sequence of 3 nucleotides, in the messenger RNA. Then we have ribosomal RNA, which is a part of the ribosome. Ribosomes are the structures that carry out translation. They are composed of ribosomal RNA and many different proteins. Each ribosome consists of two subunits, one small and one large, that contain various building, binding, and catalytic sites needed for synthesis of a protein. Here's another view of a messenger RNA molecule. Notice it's single-stranded, and it has uracil as the base instead of the T base you would see in DNA. And this slide is just showing the 4 nucleotides that you would find in RNA, and also RNA has the sugar ribose instead of deoxyribose. Let us review. Question 9. Why is RNA significant to a cell? Choose the correct answer. Okay, the final segment we're going to talk about in chapter 13 is how is the information in messenger RNA ultimately used to make a protein. This slide gives an overview of the entire process of creating a protein from the information in DNA. Genetic information is transcribed into RNA. Then it is translated into protein. DNA directs protein synthesis in a 2-step process. Information in a DNA gene is copied into RNA in the process of transcription. This occurs in the nucleus of a eukaryotic cell. Messenger RNA together with tRNA, amino acids in a ribosome, synthesize a protein in the process of translation of the genetic information contained in the messenger RNA. The translation occurs in the cytoplasm of eukaryotic cells. Let's watch an animation that gives an overview of gene expression.
The sequence of nucleotide bases in DNA carries genetic information in units that are called genes. Structural genes encode the information for specific proteins. To create a protein, a gene must first be transcribed into a sequence of nucleotide bases in a messenger RNA molecule. The messenger RNA sequence is then translated into an amino acid sequence of a protein. This sequence of amino acids in a protein molecule determines the shape and chemical characteristics of the protein. Thus, each structural gene specifies a specific protein in the cell that carries out a specific function based on its chemical characteristics and molecular shape. This function of the specific protein gives the cell and the organism the specific trait coded by the gene.
Question 10. Where does protein synthesis occur? Choose the correct answer. This figure just highlights some of the differences between transcription and translation. Transcription is synthesis of RNA, and translation is synthesis of a protein. Transcription occurs in the nucleus, and translation occurs in the cytoplasm. This figure is showing the genetic code that is used to build a protein. The genetic code uses 3 bases to specify an amino acid. The genetic code translates the sequence of bases into nucleic acids, into the sequence of an amino acids in proteins. DNA and RNA contain 4 different bases. Given that there are 20 amino acids but only 4 bases, statistically the smallest number of bases that could combine to yield a different sequence for each of the 20 amino acids is 3. A 2-base code would only produce 16 combinations. A 3-base genetic code has the potential to create 64 combinations. Bases in messenger RNA are read by ribosome in triplets called codons. The genetic code is usually written in terms of the base triplets in messenger RNA. There are 1 stop codon and 3 stop codons.